This is Bio 1 for Professor Yeargain's class, Monday, Wednesday, Friday, 1:30 to 2:20. If you have Yeargain for any other section, that's fine as well. It's Bio 1, just another reminder. So if you guys found my exam review, this is where it is, ucfstudyunion.wordpress.com. You can visit that. It's under the biology tab. So let's get started. All right, so who can tell me the number of what differs in isotopes? Yep. Yeah. The number of what differs in isotopes? Neutrons, great. So what happens if you change the number of protons? Good, it's a different element. All right, what is the mass number of an atom? What was that? Yep. Protons and neutrons. It's just something you guys have to know. All right, number 3. What are the 4 essential elements of life? C, good. Anyone remember the acronym for this? HONK, good. What else can you determine from this? Has to do with bonds. So you can find the number of bonds each of these forms with another element. So H is 1, O is 2, nitrogen is 3, carbon is 4. Everyone good so far? Which of the following describes an atom that has lost an electron and is positively charged? Cation, good. Anions are negatively charged. All right, good. What kind of bond will form in the following molecules/compounds? Polar, nonpolar, nonpolar covalent, ionic, or hydrogen? So what is CO2? Polar covalent? Okay, so let's draw out CO2. So there's dipoles going in both directions, right? So is it polar or nonpolar? It's nonpolar. Good. And is it covalent, ionic, hydrogen bond? Covalent. What about H2? What is it? So it is covalent. It's also nonpolar. What about BAS? Ionic, good. So covalent is between 2 nonmetals, and ionic is between a metal and a nonmetal. Does it make more sense? Yeah. What about water? Hydrogen, okay. So let's just say it's hydrogen. But what type of bond is within H2O? It's a polar bond, right? If we draw it out. There's these two dipoles. So it's not a hydrogen bond. What's a hydrogen bond? Is it within one single molecule? No, right? It's between two different molecules. So what is H2O bonded to NH3? What type of bond is that? What is it? Hydrogen. So it looks something like that, over and over again. Does it make sense? What characteristics below are those of a hydrogen bond? Are they weak? Yep. Are they extremely important to the specific properties of water? No, they're not. All right, well, let's just say this one's wrong, maybe. Are they temporary? Yes, they are, right? So even if you guys didn't know that this one was correct, you already have two, so it's all of the above. All of these are true of a hydrogen bond. Everyone good so far? All right, a small sample of NaCl is added to water to create a new mixture. Which of the following is the solvent? What is it? The water. Good. So what's the solute? What is it? Good. And the solution? What is it? Yeah, mixture, solution. Everyone good on these terms? All right. Which of the following is most hydrophilic? So does hydrophilic mean water-loving or water-fearing? Loving. Good. So we're looking for something that's polar. Do you see anything out of these four? Which one of these is polar? Anyone know? What was that? A. Good job. So CH4 is a non-polar molecule. We draw that out. It's a non-polar molecule. H2, we already said when we run number 5, it's non-polar. Fats and oils are always hydrophobic. So what does pH measure? Is it the OH minus concentration? No, right? What is that? What is the OH minus concentration? POH, good. So which one is it? C, good job. All right, so now we're dealing with the concentration of H+. So which one of these is an acid? Let's find the pH of all these. What's the pH of the first one? 14, right? Remember, if it's just 1 times something, it's always just this exponent, the absolute value of that exponent. So what's B? For B. 7. What's C? 9, and D is? So remember, the scale is 1 through 14. 1 through 6 is what? Acidic. 7 is? Good. 8 through 14 is? Good. So what's the answer? D. Good job. So what differs among isomers of the same element? Is it the number of protons? No, right? We already said that if you change the number of protons, change the element. Is it the number of neutrons? No, right? That makes it an isotope, not an isomer. Is it the arrangement of the molecules? Yes, right? Good. So what are the different type of isomers? Awesome. So remember, structural is the same formula, but they're just arranged differently. So you might have a carbon chain that looks like this. Remember, these points are all just carbons. So we have 7 there. And you might have something that looks like this. 1, 2, 3, 4, 5, 6. So they have the same formula, but they're just drawn out differently. Does anyone know what geometric isomers are? Perfect. Cis and trans, right? So remember that these deal with double bonds. So the relationship of these two is what? Are they cis or are they trans? Trans, good. Cis would be on the same side. Number 12, which of the functional groups are present in the following molecule? So you guys can just call them out. Phosphate, good. What was that? Awesome. Carbonyl right here. I see one more. Yeah. Yeah, sorry. So what is the last functional group here? Methyl. So we have B, D, and E. Everyone good? Which of the following refers to the process in which a water molecule is added to separate monomers? C, good. Hydrolysis. So remember, when you're having a hydrolysis reaction, you are doing something like this. Right? These circles are the monomers. And you are breaking down these two. These two combine, and then you form this, right? So this is hydrolysis right here. Everyone good so far? Where's what? So the H and the OH combine. They get like released and they combine to form water. This part right here? Yeah, so these two leave and they combine to form water, and then these two now link up. Okay. Does it make sense? Yeah. Any other questions? All right. Which of the following does not have a specific polymer? Anyone know? A? All right, so what is, let's see if we can list them. So the polymer of nucleic acids is nucleic acids. What about proteins? What are proteins made out of? Amino acids, good. What about carbs? Saccharides, right? Saccharides of some form. So then it's lipids. Lipids don't have a specific polymer. All right, number 15. Which of the following represents a proper ratio for carbs? A, okay. You see any others? D, right? So the ratio is C, H2O, N. So N can be like any number, and then you multiply it out to everything. So in this case, what would N be? 4. Everyone good? Which of the following is a type of polysaccharide that is extensively branched and made by animals? Is it glucose? No. What about starch? It's not starch, but starch is branched and coiled. It's also digested by humans. Is it glycogen? No? All right, maybe not. Is it cellulose? No, cellulose is straight, right? And it's not branched. Is it chitin? No. So yes, it is glycogen. So yeah, this is something you kind of have to memorize. I think she went over this in one of her PowerPoints early on. Everyone good so far? All right. What type of lipid is shown in the picture below? Unsaturated, right? So even if you didn't know, it has to do with saturation. So what makes you say it's unsaturated? Awesome, right? It's double bond. So it's missing 2 more hydrogens that would make it saturated. Does that make sense? All right, true or false? Some types of lipids will mix with water due to being hydrophilic. True or false? False, right? Lipids are what? Hydrophobic. Good. Which of the following is not a component of an amino acid? What is it? Carbonyl. Good. So the basic structure of an amino acid is something like this. Has an R group, hydrogen, carboxyl, and an amino group. Let's see. Whoa. Is that better? Yes. Okay. All right, number 20. Which of the following molecules orient themselves into a sandwich-like membrane because of their hydrophobic components within the membrane? What was it? D. Good. All right, so now I wanted to kind of break into chromosomes and genetics before we move on. I know a lot of you guys struggled with that in my session, so I'm just going to kind of dive into that for a second. Everyone good so far? Anyone have any questions, 1 through 20? All right. So out of these two chromosomes, which chromosome is replicated? B, right? Well, here it is replicated, right? So it has two sister what? Two sister chromatids. So after what phase does it look like this? S phase, good. What's the difference between a diploid cell and a haploid cell? Diploid is 2, right? So diploid has its homologous pair. What about haploid? Does it have its homologous pair? No, right? What is it? In animals? No, gametes. In gametes, yes, good. What other process is a haploid process? Mitosis or meiosis? Meiosis. What's a diploid process? Mitosis, good. And is mitosis for somatic cells or sex cells? Somatic, good. So now let's see where you guys stand with this stuff. So somatic cell, oh, did anyone need this for a second? All right, so a somatic cell has 4 chromosomes, so its 2n number equals what? 4, good. So how many chromosomes and chromatids are present per cell at, start with G1 phase? How many chromosomes are present in G1? 4. How many sister chromatids? 8. So has it gone through S phase yet? No, so then how many? So there's 4 chromosomes, right? It kind of looks like this. Do these have any sister chromatids? No, so then how many? You just said there's no sister chromatids, so then what is it? 0, right? Come on, guys. All right, what about S phase? How many chromosomes? 4. Now how many sister chromatids? 8, right? 1, 2, 3, 4, 5, 6, 7, 8. G2 phase? Same thing, right? What about prophase? Has anything changed? No. What about metaphase? Same thing. Anaphase? What is it? Well, how many chromosomes do we have? 8. Anyone else have a different number? 4. Okay. So let's start out with prophase. So this is a cell in prophase. 2n equals 4. What happens in metaphase? They line up on the metaphase plate. What else happens here? Okay, so we have stuff with centrosomes. Does something else attach? Spindle fibers. What do they attach to? The centromere, but specifically what on the centromere? Starts with a K. Kinetochore, good. So then in anaphase, they get pulled apart. So now we're at anaphase. So now looking at this, is this 4 or 8? 8. How many sister chromatids? None. Good. And then telophase and cytokinesis is basically this like splitting in half. So how many per cell? How many chromosomes per cell? Sister chromatids? None. So as you can see, we started with 4 chromosomes, 0 sister chromatids, and we ended with the same thing. If you guys don't get all this now, that's fine. You guys can come up and take pictures of it. Everyone good? Anyone have any questions? All right, so now let's flip the script and try it with meiosis. It's the same number of chromosomes. How many chromosomes do we have in interphase? 4. Good. How many sister chromatids? 0. How many tetrads? 2. Okay. So have we formed the tetrads yet? No, right? S phase. Are they replicated now? Right? They have. So we have 4 chromosomes. How many sister chromatids? 8, right? We have 8. How many tetrads? We haven't formed them yet? Oh, but we have. So we have 2 tetrads. So the way to remember how many tetrads you have is basically your haploid number. So if we have 4 chromosomes, we have 2 tetrads. If we have 8, how many tetrads will we form? 4, right? What about G2 phase? How many chromosomes? 8? Okay. So has anything changed from the S phase to G2 phase? No, so it's all the same. What about prophase 1? Has anything changed? Metaphase 1? Not yet, we're getting there. Anaphase 1. Yes, right? So how many chromosomes do we have? 2? So let's draw it out. So what important event happens in prophase 1? Crossing over, right? So what are these sites right here? What is it? Yeah, recombinant sites. What else are they called? Starts with a C. Chiasma, right? Good. So here they're exchanging genetic information, which is really important for when you recombine with another sex cell to form a human. So that's prophase 1. What happens in metaphase 1? They line up. They line up on the metaphase plate. Fibers attach. What about anaphase 1? They're getting pulled apart. So how many chromosomes do we have? 4. How many sister chromatids? I heard 2. 8. Do you guys see any tetrads? No, right? From this point on, are we going to see any more tetrads? No, right? They've been pulled apart. So here they were tetrads. They're no longer associated. Telophase and cytokinesis 1, how many chromosomes? Per cell? 2. How many sister chromatids? Yeah. Yeah, I'm assuming on the test it would say per cell. Yeah. What are tetrads? Good question. So this would be a tetrad, right? It's an association of 4 sister chromatids. And right here, they exchange information. So they only do this in S phase until metaphase 1. Does that make more sense? Okay. So how many sister chromatids in anaphase 1? Oh, wait, so, sorry, we already did that. Telophase and cytokinesis. All right, so now do we go through interphase again? Got some yeses, got some nos. Which one is it? No, right? We don't go through interphase again. All right, so I'm not really going to break down meiosis 2, and why do you think I'm not going to? What process is this similar to? Mitosis, right? It's essentially mitosis. So it's haploid now, but it stays haploid. So the second part of meiosis is a diploid process, but the entire process is haploid. Yeah? No, so they're not tetrads in mitosis. Only in meiosis, yep. So let's go through it really quick. Prophase 2, how many chromosomes? 2. Sister chromatids? You had it. 4. Metaphase 2. How many chromosomes? 2. How many sister chromatids? 4. Anaphase 2. How many chromosomes? 4, right? So these have been pulled apart in anaphase 2. So when they're pulled apart, they're all still in the same cell. They haven't split yet. So we have 4 chromosomes. How many sister chromatids? 0. Yeah, do you have a question? Okay. So I'm just going to skip down to like mitosis, meiosis, metaphase 2. So it looks like this, right, per cell. So then these attach, right? And then they get pulled apart. So we have 4 now. And then telophase and cytokinesis, how many chromosomes? Per cell? 2. Sister chromatids? All right, everyone good on that whole process? Yeah. Yes, I will try to post them all on my website. If not there, then I'll have a link to where they are. Yeah. Is that good? All right, so let's hop back into the multiple choice. So we have like an hour and a half left, and we have 60 more problems. So from here on, if anyone has specific problems, you can just call them out and we'll just address those. So does anyone have a specific problem they want to go over? Just in case we don't get through everything, I want to hit the problems you guys really want to go over. All right, well, if you guys have a question, let me know. Until then, I'm just going to keep going. What type of structure shows a linear chain of amino acids joined together by covalent bonds? The linear structure. Primary, right? They're linked by peptide bonds. They look kind of like that. Does that look familiar? So what about secondary? Hydrogen bonds, right? So what are the 2 shapes we can form here? Beta sheet and alpha helix. Good. What about tertiary? R groups. Good. So remember, an R group can be literally anything. It's just the different part of the amino acid. This is what differs in amino acids. And lastly, what about quaternary? What is it? I can't hear you. Yes, yes, interaction between multiple proteins. Good. Yep.
Explain more on like what actually is kind of
What do you mean?
specifically like what it means.
Okay, so you start with primary.
Sure, thank you.
So primary, we have the peptide bonds, right? It's just a linear sequence. Each of these is a specific amino acid. And these linkages right here are peptide bonds.
Okay, so as it goes up, like primary, secondary, is it being built or?
Yes, the protein's kind of being built and it's getting more complex.
Okay.
And do you remember what structure you can call it a protein? Primary, secondary, quaternary, tertiary, quaternary. So when does it take its 2D shape? Or 3D shape, sorry. Tertiary, okay. So the alpha helix, is that a 2D shape or a 3D shape? 3D shape, so it's the secondary. So the hydrogen bonds are basically when it kind of wraps around or when it's a beta-pleated sheet, they interact with each other to form these shapes. Does that make sense? Yeah. Okay, and then tertiary is the interaction between the R groups. So the way these are aligned in the beta-pleated sheet and alpha helix, these R groups interact. So the R groups are the varying chains. And let's see. You remember the structure? So this is the R group. So the R group can be anything. It can be a super long hydrocarbon. This is what makes the amino acids different.
There are 20 of those, right?
Amino acids?
Yeah.
Yeah. And then quaternary is multiple different proteins, so fully assembled proteins all interacting together. I don't think you guys need to know any more details than that, really, for this test. But does that make more sense?
Yeah, great.
Okay. Does anyone have a specific question yet? All right. Which of the following is not a pyrimidine? What is it? A. So a clever way to remember this is for pyrimidine, cut the pi. Cytosine, uracil, thymine. And then for purine, I think of pure as gold. Does anyone know what gold is on the periodic table? Ag. Yeah. So we have Ag. And then which of these is a single ring? Pyrimidine or purine? Wait, what? Oh, is it? That's not good. Is it really? Yeah. Oh, man.
There's another one. It's like all gods are pure.
Okay. So forget that. Pure is silver then. Okay, so which one of these, pyrimidine or purine, is a double ring? Purine, right? Everyone good? Yeah. All right. If a DNA sample was composed of 40% adenine, how much guanine is present? So this is whose rules? So 10, yeah. It's Chargaff. So if we have 40% adenine, how much thymine do we have? 40%. So how much is left? There's 20%, right? So then 10% has to be guanine, and then the other 10% has to be good. All right, so deoxyribose and ribose refers to the blank in DNA and RNA. The sugar. Anyone have any questions on that? Does anyone have any specific questions yet, if you guys are flipping through it? No? Just keep going? All right. If a DNA strand has a sequence, what would be the polarity and sequence of the other DNA strand? So if it starts in the 3' right here, what would it start with on the other strand? 5', right? So what's the answer? All right. When comparing plant and animal cells, which of the following is not a unique characteristic to plants? Cytosol, right? That's not unique to plants. A cell wall is. Chloroplasts are unique to plants, and so is a central vacuole. Which of the following terms refers to the selective barrier that controls what can enter and leave a cell and contain cytoplasm? Membrane, right? Plasma membrane. What type of ribosomes? Intermediate filaments? Yeah, so I don't think she really went over intermediate filaments too much. She hasn't, right? So I don't think you guys need to worry about that for this exam. And then these two are what? These are just organelles, right? Yeah. Which type of ribosomes usually make proteins that are to be sent out of the cell? Free, okay. So free proteins, what type of proteins do they make? They're for within the cell. What about bound ERs? I mean, bound ribosomes, what are they bound to? The ER, which ER? Smooth or rough? The rough, right? These are exported out of the cell. Everyone good? Any specific questions yet? Nope. All right. Which organ would you expect to have the most smooth ER due to its detoxifying properties? The liver, right? So the liver partakes in detox and the smooth ER. If you have more smooth ER, that means kind of like a heavy drinker. So liver. Which organelle contains enzymes that can digest organelles which no longer work? Lysosomes. Good. Any questions on that? All right. True or false? Mitochondria contain a single membrane and are the site of cellular respiration. False. So are they the site of cellular respiration? Yep. Do they contain a single membrane? No, right? They have many. Phagocytosis refers more specifically to what type of lysosome breakdown? What is it? Food, right? So phagocytosis is cellular drinking or eating? Eating. Does anyone know what cellular drinking is? Penocytosis, good. Everyone good? Cellular drinking, okay. So when a macromolecule is brought in, the macromolecule is kind of dissolved into the fluid of the cell. So it's cellular drinking, so it uptakes all of that fluid. And then cellular eating is because they still stay kind of big molecules within the cell. Any other questions? I'm not sure what chapter it is, but I think she went over this. In the beginning? Okay. Sorry about that. I don't know what chapter. What role does O2 play in aerobic respiration? The final electron acceptor. Good. Definitely doesn't play no role. And the rest of these just aren't, right? Everyone good on that? All right, what does it mean for the cell membrane to be amphipathic? C, good. Both A and B, right? Remember the phospholipid bilayer kind of looks like this, right? In a very simplified drawing. So this head right here, is it hydrophobic or hydrophilic? Hydrophilic, right? Because these heads are what's exposed to the fluid within the cell. What about the tails? Hydrophilic or hydrophobic? Phobic. So while we're talking about this membrane, what does cholesterol do? What is its function? Recognition, and there's one more thing.
To adjust the distance in between.
So it can affect the packing, which essentially results in regulating fluidity of the membrane. What is it? Is that what? Didn't I say cholesterol? Cholesterol regulates that.
Oh, okay.
Wait, what did I say?
Place of carbs do that.
Did I? Oh, okay. No, so carbs are also for cell-to-cell recognition. All right, does anyone have any specific questions yet? All right. Which of the following is characteristic of an integral protein? Do they bind to the hydrophobic region of the cell membrane? Okay. Can they be transmembrane? Yes, right? They can go through the entire membrane. So it kind of looks like this. You have an integral protein like that. You can also have one that goes all the way through. Will it penetrate the nonpolar region of the cell membrane? Yes. So then it's what? It is all of the above. So what are the proteins that lie like around here? What are those called? This one. What is it? Peripheral, good. So the way I remember that is they lie on the periphery of other proteins. Everyone good? All right, so we just talked about this. What is penocytosis? Cell drinking, okay. Is it anything else? D. So what are these two again? What is it? Phagocytosis. All right, so looking at this image right here, we have a solution that's 80% water, 20% NaCl, and then the cell is 80% NaCl, 20% H2O. So the cell to the solution, is it hypotonic, hypertonic, or isotonic? Iso? Okay. Anyone else think something different? Hyper. What else? Could it be hypo? So it's hyper, right? Because we have 80% NaCl here and 20% NaCl here. So which way is water going to flow? Is water going to flow from the cell to the solution or the opposite? What is it? Into the cell, right? So is it going to lyse or is it going to shrivel? It's going to lyse, right? It's going to like blow up. Does that make sense? Another way to remember it is if you're talking about the solution is hypotonic. O is in H2O. So more water. All right, what is the purpose of the electron carriers NADH and FADH2? Yeah.
Know the different wording for plants and animal cell. Well, there's one for turgid and there's one for lyse.
Yeah, okay. You should know those. So what is turgid? Is that the same as like lyse? Oh, sorry. Okay, and then what was the other one you said?
And there's shrivel and shrivel, which is the same.
Shrivel is shrivel, or there's another term. It starts with a C. Crenate. All right, good question. So any more questions? Yeah. I don't know. I mean, I don't see the test, so I can't really tell you, but I would be prepared for it. Yeah. So turgid is the same as what? Bursting. What is the purpose of electron carriers NADH and FADH2? What is it? B, right? Electron transport chain. Specifically where? There's complexes, right? So NADH delivers it to what complex? What is it? First one. What about FADH2? Second complex. So while we're talking about that, what builds up? Concentration of what? Hydrogen, right? Where? Outside and what? So it's not the cristae. Yeah. Intermembrane space. Yeah. And it goes down through what? What is this enzyme here called? ATP synthase. Into the what? The mitochondrial inner membrane. So there's a space in between. That's the inner membrane, but it goes through that. So what's inside the inner membrane? What's past that? I heard it just now. The matrix. So does this process generate a lot of ATP? Yeah, a lot, right? Does anyone know a specific number for year gains class? Oh, yeah, sorry. 28, right? So 28 to 34, that's like the range. If anyone here isn't in year gains class, these numbers are going to vary. All right, which of the following is the substance that is produced by the oxidation of pyruvate and then fed into the citric acid cycle? Acetyl-CoA. That's not obviously pyruvate, it's not glucose. What was the role of O2 again? Final electron acceptor. After it accepts that electron, it's now what? After it accepts that electron. What is it? Water. Good. And if you guys need me to move this up at any point, just call me out. I'm not going to keep looking back. Does it make sense? Everyone good? All right, so we're about halfway through. Does anyone know what time it is? What is it? 9:30. Okay, so why don't we take like a 2-minute break, and then we will pick back up. And then in that time, you guys can like think of specific questions or 42. So if you guys want to think of specific questions, we can go to that first, and then I will obviously be posting the answer key to this sometime tonight or tomorrow. And if you guys have questions, you can come up here too. What is it? Oh, yeah. We have, you can take pictures of this if you want. It's a cellular respiration summary. All right, let's get started again. So does anyone have any specific questions? Nope? All right. So number 40, photosynthesis takes place in the membranes of A, thylakoids. So where does the Calvin cycle take place, the synthesis part? The stroma. What about the photosystems? How many photosystems are there? 2, right? Where does light pass through first? Photosystem 2 or 1? 2, right? Does anyone know the wavelength? Is it P680 or P700? 680. Then we go to photosystem 1, which is P700. What are photons? Okay, particles of light. Yeah, that was like a very physics definition. No, good job. And they contain energy, which is absorbed in the light harvesting complex. Everyone good? All right. What type, did you have a question? No. What type of phosphorylation is responsible for the majority of ATP production during cellular respiration? What is it? B, good. Oxidative phosphorylation. So that's this process right here. All right, which of the following occurs in the stroma of the chloroplast? Calvin cycle, good. All right, which of the following is not a product of the light-dependent reaction? Sugar, right? The rest of these are products of the light-dependent reaction. Does anyone know what goes into the light-dependent reactions? What goes into the light-dependent reactions, or the photo part? What is it? ADP, good. What else? CO2? So CO2 doesn't go in. But there is a form of energy. What type of energy? NADPH. Actually, no, the H plus isn't there, so it's just NADP plus. And then there's light energy, right? And then one more thing. What do you give a plant for it to grow? Water. What comes out? Good. What do we breathe? Oxygen. And then one more thing. ATP. So this ADP gets phosphorylated, becomes ATP. Everyone good? Yeah. So which one is the Calvin cycle light dependent? No, it's not. So why is the Calvin cycle kind of light dependent? Because it relies on the products of the light dependent reactions. So once it uses all of that up, then it can't keep going. But no, the light dependent reaction is the photo part of photosynthesis. Any specific questions yet? No? What is it?
Why is it CO2 as whatever it goes into the light-dependent reaction? Is it because it goes into the Calvin cycle?
It goes into the Calvin cycle. Yeah. Yep. All right, if a new found animal has a haploid number of 60, what is its diploid number, and would the diploid number be the number of chromosomes found in gametes or somatic cells? So its haploid number is 60. What's its diploid number? 120. And is it 2n equals 120 or n equals 120? What is it? 2n. And are these in somatic cells or gametes? Good. All right, so in a karyotype, which is basically when they list out all the chromosomes, how many pairs of autosomes does a human have? 22, right? What's the last one? The sex cells, right? So you have either, this doesn't really look too accurate, XX or XY. All right, which phase of mitosis do sister chromatids separate? Anaphase, right? So you guys remember, where'd it go? Right, so there's sister chromatids here, and then they separate an anaphase to form just double the number of chromosomes. All right, which choice below properly matches the stage of mitosis with its description? So does telophase result in the formation of two nuclei? Yes, but let's read the rest. Metaphase, the cell elongates and cohesion proteins begin to denature. No, when does that happen? So I heard prophase. So let's just keep going. So prophase, chromosomes line up on the metaphase plate. Which one is this one? Metaphase. All right, and then when does chromatin condense and the microtubules form? Prophase. Good, so the cell elongates and cohesion proteins begin to denature where? Anaphase. So the answer is A. Everyone good? All right, true or false? Gametes are diploid cells. Okay, we can stop there, right? Are gametes diploid? Okay, good. Which of the following is a complex of proteins at which the mitotic spindle will attach to the chromosomes, one microtubule from each side? D, right? Kinetochore. Everyone good? All right. The mature form of a newly discovered species of eukaryote contains 12 chromosomes and exists in the diploid state. Its 2n number equals 12. In this new species, how many chromosomes are present in a mitotic cell immediately after anaphase? What is it? 24, right? So they've temporarily doubled, and they do this so that when telophase happens, they split back to their original number. All right. During which of the following mitotic phases will a cell be haploid? None of the above, right? It's always diploid throughout the mitotic process. True or false? The mitotic phase is where the cell spends most of its time during cell division. False. Where does it spend most of its time? Interphase. If you were to draw it out, it's the mitotic phase. What is this first one? G1. Second one? And this isn't really that proportional, but S. Last one. What stage does the cell enter if it doesn't want to divide? If it does not want to divide, the cell enters what stage? G0. Everyone good? All right. Yeah. So during which of the following mitotic phases will a cell be haploid? So if we look here, this is mitosis, right? So at no point has it become 2 chromosomes in the cell, which would be the haploid part. So it's 4 here, 8 temporarily, and then it goes to telophase and cytokinesis, resulting in 4 each. So yes, it's, which question was that? Yeah, so none of the above. It's never haploid. It never has 2 chromosomes per cell. Does that make more sense? Okay. All right, when referring to the paternal and maternal versions of chromosome 3, we can call them blank in relation to each other. Yeah, you have a question?
Yeah, sorry, just from another one. When it goes to G0, that's before or after S phase?
So G0 can occur in any phase, really.
Oh.
Yeah, so G0 is just when the cell, it can either fail to meet a checkpoint or cell realizes it doesn't need to divide anymore. So it just enters that non-dividing stage. Yep. So what's the answer to 53? Homologous, right? So why aren't they sister chromatids? They're not the same, right? They're not like completely identical. They're not attached either to one single centromere. And they're not identical. All right, 54. Scientist is interested in studying the event of crossing over, recombination, in mouse gametes. She wants to label some of the mice with fluorescent dyes to better visualize it. During what phase of the cell cycle should she look at these fluorescent DNA strands to most effectively study recombination? So she wants to see the recombination event. Where does that happen? What is it? Prophase 1. Yeah. Yeah.
For that, like, is there a difference between homologous and all these pairs?
No.
Okay.
Good question. Everyone good on why 54 is prophase 1? All right. Which of the following statements regarding mitosis and meiosis is correct? Does mitosis occur only in prokaryotes? No. Does it result in higher genetic variability than meiosis? No. Is there any genetic variability from mitosis? No, it's the same thing, right? So can it be C at this point? Yeah? Did someone say yes? No. In the human body, mitosis occurs in somatic cells, whereas meiosis occurs in sex cells. Yep, that's true. So it's D. Which of the following is true of both meiosis 1 and 2? So do they both produce haploid cells? They do, right? Technically, the whole process is a haploid process. Sister chromatids separate in anaphase of meiosis 1, but not in meiosis 2. No, right? What separates an anaphase of meiosis 1? The tetrads. Tetrads separate. So what separates in meiosis 2? Sister chromatids. Does the G2 phase precede both? No, it only precedes what? Meiosis 1, good. Both include crossing over. That's not true. When does crossing over occur? Prophase 1. Sorry, I know I'm like drilling that into your head, but okay, I guess there's two of the same thing. So yeah, A. Cell has 40 chromosomes during G1 phase. How many sister chromatids does that cell have during prophase 1 of meiosis? What is it? It's 80, yeah. Does that make sense? So we still have 40 chromosomes, but we now have 80 chromatids, sister chromatids. A human cell has 46 total, or 23 pairs of chromosomes. Following mitosis, the daughter cell would each have a total of blank chromosomes. So for the first part, how many chromosomes? (46). After meiosis 1, the two daughter cells would have how many chromosomes? Is it still 46? They went through meiosis, right? 23. And then the last part, after meiosis 2, still 23 chromosomes. Everyone good? All right, which of the following is a source of genetic variation meiosis? Independent assortment. What about random? Okay, yeah, all of them. Crossing over is definitely a source of genetic variation, and so is random fertilization. All right, which of the following explains why XXX females usually go undiagnosed? So is their life expectancy very low? No, not really. All but one X chromosome can be inactivated? Yes, right? What are those inactivated chromosomes called? What was it? Bar body, right? We don't have the technology. No, we do. It's the Y chromosome that creates the observable defect. No. And nope. All right. In Mendel's F2 generation of the purple and white flower crossing, the dominant to recessive ratio was what? What is it? D. Okay, so D would be a dihybrid. But we're only talking about one trait here, right? So it can't be D or A. So does anyone remember what the parental generation was? What he crossed? It's dominant purple and dominant or recessive white, or the other way around. So let's just say A. So these are the parental generations. So what is the resulting AA? What generation is that? 2? So not F2, it's not F2 yet. It's F1. It's the first filial generation. So then the second generation was a cross between two of these. So how many of these are going to be expressing the dominant trait? 3. And recessive? 1. Yeah, I know this is kind of a tricky question. I know you guys probably didn't remember this stuff, but it could be on there. Does that make sense? All right. Where two alternatives for a trait are tall and short, and tall is dominant, the phenotype of the heterozygous individual would be expressed as E, right? What are all of these? Genotype. Good. And then this is phenotype, which is what you see. All right, in the cross MM, big M, little n, and the opposite, what proportion of the resulting F1 would be homozygous dominant for both genes? None, right? We wouldn't even need to work this one out. Because if you have big M here and little m here, are you going to have any fully dominant? No. Same for the opposite. Does anyone want me to work this out and see what results? No? Okay. Which of the following describes multiple genes affecting one trait? A, right? Polygenetic, so polygenic, many genes. What is epistasis? Yeah, when one affects the other, right? So it kind of masks one trait. X inactivation results in the formation of what? Bar bodies. And we will get into pleiotropy in a minute. So black hair for horses, big B, is dominant over brown, little b. However, when a homozygous dominant haired female is crossed with a homozygous dominant male, few offspring show a whitish tan color due to a third gene that is present. What could be the possible explanation? Epistasis, right? So none of these even code for white. So the only way something that could result in white is something masking both of the effects of the other alleles. Does that make sense? All right. Marfan syndrome, a dominant single gene defect, is characterized by lens dislocation, long limbs, spindly fingers, a caved-in chest, and a weakened aorta. This is an example of, what is it? Good. So it's one thing that affects many different traits. That's why you have all of these unfortunate problems down here. So that is pleiotropy. Yes. Good example. A homozygous red and homozygous white flower are crossed, and the offspring are all pink. What is this an example of? Incomplete dominance. Good. What would complete dominance look like if this was a complete dominant trait? It would be like red with white spots, or the opposite, right? They're both expressed. Or sorry, no, that is codominance. So another way to tell that this is an incomplete dominant trait is it would be written as homozygous red would be CR, CR, and white would be this. So what would the resulting generation be? RW, good. That makes sense? All right. So the way I remember it is codominant is both are expressed. So they're both expressed in like full effect at the same time. So white is fully expressed and red is fully expressed. That's why they both show up. Whereas incomplete is none is fully expressed. They're both kind of blended. All right. What is the purpose of topoisomerase during DNA replication? C, good. So what holds the DNA polymerases in place during replication? Good. Single-stranded binding proteins. And so even if you didn't know that it cuts the phosphate backbone ahead, you guys should remember that it prevents it from getting overwound. Good? All right. Which of the following refers to the correct model of DNA replication, where a newly synthesized strand of DNA is half the parent strand and half is newly synthesized? Semi-conservative. So it should look something like this. This is the parent strand, and then that's the new strand. All right, true or false? DNA polymerase can only add nucleotides to the 5' end of the DNA strand. True, false, which one? False, right? So DNA is synthesized in what direction? 3 to 5 DNA is red. 5 to 3, it's synthesized. So on the template strand, it starts at the 3' end of the DNA strand and works its way, reading the template. So it's the 3'. All right, true or false? The two sides of the double helix structure of DNA are held together with weak hydrogen bonds. True, right? So hydrogen bonds are weak. We went over that, and double helix structure is held together by hydrogen bonds. All right, 72. DNA is the genetic material of all living organisms. Which of the following describes the difference between prokaryotic DNA and eukaryotic DNA? So let's go through the first one. Eukaryotic DNA is antiparallel, prokaryotic is not? No. Prokaryotic DNA uses uracil, while eukaryotic DNA uses thymine. No. Where is uracil used? RNA. Prokaryotic DNA is haploid while eukaryotic DNA is diploid? No. Prokaryotic DNA is circular while eukaryotic DNA is linear. All right. DNA polymerase and RNA polymerase have similar functions. Which statement is an incorrect description of these enzymes? So does RNA polymerase require a primer? No, right? It just goes straight ahead and does it. Does DNA polymerase require a primer? Yes. So what comes in and changes those RNA to back to DNA? Polymerase. So DNA, which one? 1 or 3? 3? 3 and 1. So which one does 3 do? Does 3 make the DNA? Yeah, so 3 comes in and makes the DNA. DNA polymerase 1 comes back in and makes the RNA, or changes the RNA to back to DNA. So incorrect is this. Everything else here is correct. All right, 74. The codon chart. This one?
I want to ask, what is the other choices for?
Dispersive. Okay, so dispersive is what? Does anyone know? So she didn't talk about dispersive in class. So if she did, I would have gone over it. Otherwise, I'm not going to go over it. What about conservative? No, I wouldn't. I don't think so. But I would recommend knowing what conservative is. So what is conservative? What? Oh, knowing what conservative is. So conservative would just be this whole thing is just completely replicated just like that. It's the same thing. All right. The codon chart shows that there is redundancy but not ambiguity in the genetic code. How can this statement be explained? So one codon can code for several amino acids. Is that true? Okay. Any mistakes are corrected during the proofreading process. Got some yeses. Let's keep going. Individual amino acids can have more than one codon, but each codon only translates one amino acid. Yes, so that's where the redundancy comes in. And then D is just like not true at all. Everyone good? All right. Which enzyme is essential for transcription as functions such as unwinding the double helix and synthesizing mRNA? So I heard polymerase, so you're right. Which one? RNA polymerase. All right, 76. In eukaryotes, we make pre-mRNA that must be edited before released into the cytoplasm. How is the pre-mRNA prepared? Is there a poly A tail? Yep. Is there a 5' cap? Yep. Does splicing occur and remove introns? Yes. And exons remain, right? All of the above. All right. At which site does the large ribosomal subunit does the first tRNA bind? I think this is a learning catalytics question. What is it? The P site, right? Does anyone know what the start codon is? AUG. All right. 78. In a species of lab mice, there is a gene with 2 alleles that determines fur color, with the black allele dominant over the brown allele. There's another gene with 2 alleles that determines if the mouse is bald or not bald, with the bald E allele dominant over bald little e allele. The second gene is recessively epistatic. So what does it need to be for it to mask the first effect? Big E, big E, big E, little e, or little e, little e? Little, little, right? All right, so if you breed a black mouse that is homozygous black, so what is that? Good. But heterozygous for epistatic gene, what would that be? Big E, little e? And a brown mouse, which is what? It was also heterozygous for the epistatic gene. Good. So what are the possible gametes we can form from this one? There's two. What are they? Good. What about this next one? Good. So we can make our square. So what are the expected phenotypes of the offspring? How many of these are going to be black? Okay, so it could be all of them, right? So this one's black, this one's black, this one's black, this one might be black. But remember, this one is epistatic, right? So it masks the effect of it being black. 75% black. What's the other 25%? So it's not brown. Bald. Does that make sense? Anyone have any questions? All right. If you cross a parent who is heterozygote for both gene A and B with a parent who is homozygous recessive for both gene A and B, what is the chance their offspring will be homozygous recessive for both genes? So we have one parent, heterozygote. Yeah. Actually, let me just write it out and then I'll move it up. With one that is recessive for both. So what are the possible gametes we can form from the first one? So big A, big B, what else? Nice. This one. All right, so that's it for that one. So the question's asking, what is the chance their offspring will be homozygous recessive for both genes? 25%, good. All right, a mom with blood type O. If you're blood type O, what is your genotype? II, little I, right? Has a child with a man with type AB blood. What's your genotype? IA, IB, right? So this is what we have. Type O, type A, B. So what are the possible types, blood types we can form? A or B. So that's it. These are the possible blood types, A or B. Everyone good on blood types? Yeah.
Can you go over like RH factor?
Yeah. So RH factor is what? What does that determine? Positive or negative, right? So RH positive, you have two possible combinations. What can that be? Good. So we can have positive positive or positive negative. What about RH negative? Negative negative. So this just determines if you're AB positive, AB negative, stuff like that. And they would specify if you are heterozygous for RH factor or homozygous. Does it make more sense? Okay. All right, the following pedigree shows a recessively inherited disease with the filled-in shapes indicating individuals who have the disease. What are the genotypes of the different family members? So we have 1 through 8. So 2 of these are painfully obvious. What are the 2? 4 and 8. So let's just say we're dealing with trait A and little a. So 4 and 8 are both what? Recessive, right? Good. So if they had a recessive kid, what do we know 1 and 2 have to be? Heterozygote. Good. Does that make sense why? Okay. What about number 3? Good. So big A, or, and it'll probably be written as that. What about 5? So it could be the same thing, but what do we know that makes it? So it's heterozygous, right? So they have a kid that has the trait, so it has to be heterozygous. What about 6? What does 6 have to be? And 7? Either or, right? Because we don't know if this one has kids and has a kid with the affiliate trait or not. So do 1 through 8 make sense? Okay. And lastly, in eggplants, there are true breeding deep purple eggplants and true breeding white eggplants. If you breed a purple and white eggplant, you get all lavender, pale purple eggplants. If you breed these lavender eggplants with each other, what is the expected colors and ratios? So it's asking for a cross of this, right? You're breeding the lavenders together. So what do we get? What's this first one? We have 1 purple. What are these 2? What is it? So it's not pink. It tells you here that, see, yeah, lavender. And what is this last one? White. Does that make sense? Does anyone have any questions on anything we went over? All right, well. What is it? On the exam? Probably. But thank you all for coming. And before you leave, if you guys could just fill this evaluation of me out. Won't take too long. Okay, perfect. Thank you. Yeah, you got that side? So it says, please bubble in the number provided by me. It's 26. It's on the piece of paper.
Thanks so much.
Yeah, of course. We still have 15 minutes too, so if you guys have any questions you want to come up and ask. What is it? Yeah, that's fine. Thank you. So if you guys have any specific questions, we can go over that as well. Thank you.
Also, can I see the first 5?
Yeah, yeah. You can see the entire test.